purpose of this lesson is to understand the structure of a triglyceride, and triglycerides are the main constituent of body fat in humans and other animals. They're also found in fats that are derived from plants, and while there are several different types of triglycerides that can be found in nature, one of the key differences between the different types is whether or not they are made from saturated versus unsaturated fatty acids, which we'll discuss here in just a minute. One of the things I want to remind you of, and this is something that we talked about a lot with the carbohydrate lesson, is that all of this is possible because of these three atoms. And again, as a reminder, oxygen can form 2 bonds because it has 6 valence electrons and it needs 2 additional electrons to stabilize. Hydrogen can form 1 bond. It has 1 valence electron and needs a total of 2. And then carbon comes in as a neutral atom with just 4 valence electrons. And so it needs 1, 2, 3, 4 additional electrons in order to stabilize itself. And so if you take a look at the molecules that are present in this particular picture, you see that all of them are composed of a mixture of carbon, oxygen, and hydrogen. Now, these obviously look very different from the carbohydrates that we looked at in the previous lessons, but they are made up of the same atoms. All right, so a little bit more about glycerol. So glycerol is also called glycerin, and it's a simple compound that is known as a sugar alcohol. And it is colorless and odorless. That is sweet and, I don't know what's happening here, so sweet and non-toxic. Let's see if we can get back to our video. That was fun. So it's sweet and non-toxic so it's something that you can actually taste. Now, that's the backbone of it. And when I talk about the backbone, I kind of want to just highlight a few specific things here. So it's a 3-carbon molecule. So this is kind of the backbone of it. And it has 3 hydroxyl functional groups attached. Now, you remember the hydroxyl functional groups playing a really important role in the combining of individual glucose molecules from the previous lesson. So the same thing is going to happen here, where those hydroxyl functional groups are going to come into play into building the official triglyceride molecule, which we'll do here in just a moment. All right, but before we do that, I want to kind of focus in on these fatty acids. Now, what I'm showing you here is a very shortened version of a fatty acid. These fatty acids can have hundreds of carbons long, so these tails can be really complex. And so I'm just showing you a really simple version of those here. One of the things that all of these fatty acids have in common, regardless of if they're saturated or unsaturated, is they all have what is called a carboxyl functional group. So I'm going to just write out that name because it isn't something that we have looked at yet. Sorry, carboxylic acid. So I always kind of shorten that up and say just like a carboxyl functional group, but the formal official name is carboxylic acid. And it looks similar. It looks like it's got that hydroxyl functional group in there as well. But if it has this double bonded oxygen, so if that carbon also has a double bonded oxygen to it, we can't just look at the OH part of it. We actually have to look at that whole piece there. So that's something that all of these saturated or unsaturated fatty acids have in common. And as you look at that section, I'm actually going to re-highlight that section really quick. I want you to start to think about how that could potentially connect to the glycerol backbone to form a triglyceride. And a triglyceride is a glycerol with 3 saturated or unsaturated fatty acids. So the triglyceride. Okay. So I'm going to get rid of this and just talk a little bit about the differences between saturated and unsaturated fatty acids, and then we'll go ahead and show you how these things come together. Hopefully, again, you can start thinking that through. So saturated fatty acid, which is that fatty acid there on the top, the carbon atoms are bonded to as many hydrogen atoms as possible. All the carbon-to-carbon atoms share just single bonds between them. So if you kind of look here, you've got a single bond, single bond, single bond. And then every single place that you could attach a hydrogen, it has a hydrogen. Again, carbons have to have those 4 bonds, so all of those bonds are satisfied there. So that's a saturated fatty acid. Now, this is a very common fatty acid that is found in animal fats. The straight chains that are formed here can be packed together very tightly, which allows them to store energy in a compacted form. They also have relatively high melting points, so typically you can tell that something is a saturated fatty acid because it will be solid at room temperatures. So this is the way that animals typically store their fatty acids. And then if we look at the unsaturated fatty acid, so again, it still has that carboxylic acid functional group. They all always have that. But one of the key differences that you'll notice between the two is that between the second and third carbon, there's this double bond. Well, because there's a double bond, it can hold fewer hydrogens, right? If we just do a side-by-side comparison, this one has 1, 2, 3, 4, 5 hydrogens on that, on the tail end of the molecule. But the saturated fatty acid had 1, 2, 3, 4, 5, 6, 7, right? So that's one of the biggest differences there is the number of hydrogens that it can hold. Well, because of this double bond actually forms a kink in the molecule, so it kind of makes a bend in the molecule. And what that doesn't allow it to do is to lay side by side with other lipids very nicely. And so typically these things have lower melting points and they're liquid at room temperature. This is the way that plants store their excess energy. So they use unsaturated fatty acids to store their energy. All right, so kind of looking through this, I'm going to actually get rid of this. I'm just going to sneak it down to the bottom so I don't lose it forever. And then we're going to just look at how these things link up together. So in the process of creating this triglyceride, what's going to happen is that fatty acid molecule is going to kind of move into position. And again, hopefully you guys can kind of see how this might work. It's a dehydration synthesis reaction. Actually, I'm going to do one really quick thing before I do that because I don't want to lose a copy of this. Okay, so it's a dehydration synthesis reaction. And in that, we have to remove water. So similar to what we talked about with the other, when we were linking together glucose molecules to glucose molecules, it doesn't really matter if we remove the 2 hydrogens and oxygens from this part of the molecule or if we remove it from this part of the molecule. Really, that piece of this doesn't matter. The important part is that we are removing a total of 2 hydrogens and 1 oxygen. So we'll go ahead and do that now, and we'll just kind of sneak that down here. And when we do that, that means that this oxygen is going to need to bond with something, and the carbon that's left over here, right, it only has 1, 2, 3 bonds right now, and it needs a 4th bond. So as you can imagine, what that allows this to do, oops, tried to grab the wrong thing. So what that allows us to do is it slides just into place, okay? And then what you have left over is you have 2 hydrogens and 1 oxygen, right? And again, just like in the dehydration synthesis reaction from previous lessons, water can form, right? Just a slight rearrangement of this molecule and we have a water. All right, so the same process is going to happen again. And this is going to overlap a little bit just because I didn't plan this out super well. But the exact same thing is going to happen here where this molecule is going to attach. I'm going to just draw this down a little bit lower to make it not be so bad. But we're going to do the same thing where we're going to just remove the water from here. Now I'll sneak it down there. And again, the oxygen is going to need to bond with the carbon from the fatty acid. So sneak that over into here. And again, we've got some overlap. And I'm going to just be okay with that at this point. And then the water is going to form off of that. So then we get another water. All right, and this is called a triglyceride because it'll have 3 fatty acids. So it's a glycerol backbone with 3 fatty acids. So we just have to do that process one more time. So again, we're putting together this triglyceride by using the concept of dehydration synthesis. So again, we're going to bring this fatty acid tail into place, remove 2 hydrogens and 1 oxygen. Oops, I removed a little bit too much. Hold on. So we remove that and then sneak this in to form the bond. And then rearrange this water molecule. Okay, so while this isn't perfect, like my drawing isn't a perfect version of this, I really kind of need to space things out a little bit more. You can kind of see how complex looking this molecule is. But if you can actually just break it down into the glycerol backbone, which again is a 3-carbon backbone with 3 hydroxyl functional groups, and then all the remaining spaces are just filled with hydrogen. And then you're bringing in either saturated or unsaturated fatty acids. In our example, we use saturated fatty acids. Again, in a saturated fatty acid, the only double bond you're going to see is that double bonded oxygen on the carboxylic acid functional group. And you're bringing that in and you're removing water. Now, one thing that's different from the carbohydrate lesson is that every single time that we're building, we're adding in a saturated fatty acid, we're removing exactly the same number of waters here. So in this reaction, 3 waters will be removed every time a triglyceride is formed. All right, in the next part of this video, I'm going to just build this particular molecule. It's an exact replica, just using the molecule kits, and then so you'll get to see it in person a little bit better than maybe this diagram shows you. Okay, so in this video, I wanted to just take a few minutes of your time to show you what the molecular structure should look like when we're using the model kits. So again, looking at the glycerol molecule, which is a 3-carbon backbone that has the 3 hydroxyl functional groups. When that lines up, you can kind of see, I'll just give you a bit of a view of that. One of the things that I like to do is kind of line them up so that all of the hydroxyl functional groups are in the up position, so that as I bring in my fatty acids, it's a little bit easier for me to be able to put those together. So I'm going to bring in one of my saturated fatty acids. I made this particular one a little bit longer than the one that we had in the previous note. So you'll notice that this one has 1, 2, 3, 4, 5 carbons there. And then again, kind of looking at the carboxylic acid functional group and getting a good look at that. So again, it has the hydrogen and oxygen, and then the carbon with a double bonded oxygen. So this carbon and oxygen are sharing a total of essentially 4 electrons between them, which are forming those double bonds. All right, so we're going to just go through and look at the dehydration synthesis reaction that occurs between the glycerol backbone and the fatty acid chain. Again, this is considered to be a saturated fatty acid because all of the carbons are single bonded to one another and they have the maximum number of hydrogen atoms. I'm going to show you at the very end how that allows these this particular molecule to kind of squish together to form those solids. All right, so when this carboxylic acid end comes into contact with the hydroxyl end, again, we can kind of look and see that we can remove the O and H from one end of one of the molecules and just a hydrogen from the other to link that together. I kind of think about this, it kind of reminds me of like a dog or an elephant. I don't know, I kind of think of it as a dog. And this is kind of its watering place. And so I'm going to actually just remove the OH from the carboxylic acid. So kind of exposing the, taking the nose and mouth off of my dog molecule here. And then I'm going to just remove hydrogen here. And then that leaves room for these to bond together. And so now the fatty acid is linked to the glycerol. Okay, it's not like looking any less complex than it did before in the pictures. And then this will form water. All right, so there's one. All right, I'm gonna bring in my next fatty acid and essentially do the exact same thing. Okay, so where these overlap, I'm going to remove the OH from the fatty acid, remove the hydrogen from the hydroxyl on the glycerol, and then go ahead and link those two things together. And again, you can kind of see it's getting a little bit more complicated, but hopefully you can see it's the same thing. So you're like taking these simple building blocks and just using dehydration synthesis to put them together. So even though it looks complicated, if you can just narrow down where the reaction is occurring, it's not so bad. All right, and so then the last one here, again, going to take an O and an H off of this and just the hydrogen off of here. And then to fulfill carbon's need for 4 bonds and oxygen's need for 2, we link those together, and that forms our triglyceride. And again, we have 1 additional water here, so that's a total of 3 water molecules that were formed in the process of building this triglyceride, which I would say is a key concept. And then we have our triglyceride, so it is the glycerol backbone with 1, 2, 3 fatty acids. And again, one thing I said I would show you guys is because these are single bonded, they actually can interact and they can kind of combine. And I don't want to so much say smush together, but they can. So they can occupy less space. And then these things tend to have higher melting temperatures, which means that they're typically solid at room temperature. And again, that is how our bodies store fat. I did want to show you just one more thing on this, and that is the hydrolysis reaction. So when our body is ready to actually start breaking these apart, and one thing it can do is it actually can break apart these fatty acids from the glycerol. It is the reverse process, right? So we have to bring water in order for that to occur. So I'm just gonna do in this next few moments just the exact opposite. And again, this is a hydrolysis. We're adding water to break down these molecules. So again, if I remove the fatty acid, I have to replace what is missing. So what was missing off of the glycerol molecule was the hydrogen, and then I can just reattach the nose, so reattach the OH here, and I can do that over here again. So again, take this apart, put back a hydrogen, add back the OH group, so making that hydroxyl or carboxyl group again, and then last one here, add back in the H, put this back on. And so now we have that intact glycerol backbone, and then we have our 1, 2, and 3 fatty acids one more time.